Bismillahirrahmanirrahim. Dear students, today we will start a new topic, a new subject course, which is the part of PharmD final year. It will be taught in the 9th semester and the 10th semester. So the topic name is Pharmaceutical Technology. The course name is Pharmaceutical Technology. I am Dr. Talib Hussain. I am assistant professor at IPS, University of Veterinary and Animal Sciences. First of all, I will introduce you what is why the name is pharmaceutical technology and why we have incorporated in our final year PharmD course. So pharmaceutical is a general term which is used to describe all the process in which a company or a lab or any other institute is dealing with the drug substances which are to be converted into some delivery systems or dosage forms. Dosage forms means a physical form of the delivery device. So a delivery system and its analysis, its quality control analysis, and its packaging along with its marketing. So this term pharmaceutical is a mother term for pharmacy or for pharmaceutics field on generally dealing with all kinds of medicine which have been converted to some delivery systems and they are intended for administration under the regular laws of the country. And then the technology. Technology is actually the word which have been used to optimize some things or to or in other words you can say technology is the use of some processes or a use of some techniques which are sufficient to produce pharmaceutical ingredients in a optimized form or in a form which are producing the best results and reproducible results. So let's start with the pharmaceutical. So pharmaceutical, as I described, is a term or a field of pharmacy which will deal with the dosage forms or the form in which the drug is actually available for delivery or for administration to a physiological body or known as drug delivery system, and it will include all the fields of laboratory based formulation or for a industry based formulation. And technology, as I described, is a process or a technique or a procedure which is used to optimize any system. Means a pharmaceutical system, we need to optimize the pharmaceutical process, pharmaceutical system, we will need a technology, we will adopt a technology or we used an optimization process. What is optimization? Optimization is actually the process in which we make any technique or any procedure into its perfect and reproducible form, or it will be considered as the best and most effective process or most effective technique. So optimization means whenever if a process has been optimized, it means a process will produce the same result whenever it will be reused or it will be reproducible in its actual effects. The pharmaceutical technology will be considered as a branch of pharmaceutical science which deals with all the formulation composition, the composition in which we use some ingredients in proper ratios, in proper blends to produce a batch of the formulation or to produce a number of the formulations. Then it's on small scale preparation methods. For example, in laboratory, we are using preparatory methods to prepare a mixture of different ingredients, which are known as formulation in fixed proportions to devise it into a delivery system. And on the large scale, it could be manufacturing. So manufacturing is all the process we use some instruments or some machines in which the preparatory methods will be proceeded. And then it's analysis, which is quality control. And similarly for extemporaneous and commercial manufactured products. So pharmaceutical technology, जो है वो pharmaceutical science is a branch of the pharmaceutical science जो कि आपकी स्मॉल स्केल पे और लार्ज स्केल पे दोनों तरह की मैन्युफैक्चरिंग और कंट्रोल को डील करती है ऑफ अ फॉर्मुलेशन हैविंग अ फिक्स्ड कंपोजिशन. So in this we will deal with some active ingredients which will be known as APIs and some pharmaceutical excipients which are known as polymers and the specific dosage form ingredients, specific delivery system ingredients. So the combination of these pharmaceutical active ingredient and the excipients will make a composition, and that composition in fixed proportion will develop a formulation with a method or a preparatory method to prepare a formulation. So pharmacy or pharmaceutical technology actually involves various disciplines of pharmacy. For example, we need a drug substance or a medicine for a treatment purpose. So a drug, from where we will get a drug or from where we will prepare a drug, it will be a field of medicinal chemistry. So a new drug will always come from the field of medicinal chemistry. It can be synthesized or it can be molecularly modified after synthesis also to get it feasible for a delivery system to be administered in the body. Secondly, we need its physicochemical properties. The physical-chemical properties will make it a candidate for any kind of administration. For example, if we want to administer it orally, we should have the physicochemical properties that it can dissolve or it can release from the delivery system. It can absorb from the site where it has been released, and it can be transferred to the therapeutic site at which it can produce results. So if we know its physicochemical properties, we can design it in a proper delivery system. And then for quality control analysis, as we say, we need some instrumentation or analytical techniques. For example, for its assay, HPLC techniques or spectrophotometric techniques, or the higher one can be used. And then for therapeutic quantification or to check the effects on life processes or to check its mechanism of action in which it will be moving pharmacodynamics and in which it will be eliminating or moving from one body part to another part, we will need clinical pharmacy. And for the process running into known as manufacturing or a preparation process, we will need the knowledge of industrial pharmacy so that we can use some machines or instruments to prepare large quantity of the delivery system or large batches of the system. Next, we will deal why we need this type of a subject, as we have already discussed many subjects during the our pharmacy field. For example, in the first year, we used physical pharmacy for physicochemical properties. In the second year, we used dosage form to develop a formulation at the small scale into a specific physical form. Then in the third year, we used dispensing pharmacy for extemporaneous preparations of the same delivery systems at smaller scales. And then in industrial pharmacy, we converted those small scale formulations into a large scale manufacturing process. What was the purpose? That we should add pharmaceutical technology as a subject in the presence of these 4 to 5 subjects. So the first reason that was appearing, that we were using the processes which were not reproducible. We were using process that were producing a result for the first time, and whenever a new person or a new instrument was introduced into the manufacturing plant, it produced drastically different results or different type of formulation that was not up to the standard as the first one was. So we need a optimization process so that a pharmaceutical industry which is preparing a formulation should have optimized techniques, optimized procedures, and all the equipment being qualified and validated before the time of preparation or manufacturing of delivery devices. And secondly, as the new drugs are being discovered frequently in the different countries, so our new drug development process need to be devised. So the new drug development process was devised under the heading of pharmaceutical technology. So on these basis, the pharmaceutical technology subject was actually came into shape based on the 2 basic goals or 2 basic objectives of our field of pharmaceutics. Cost-effective term is used to define a therapy in which a therapy can be utilized at a minimum cost, and every time whenever it will be administered, it can produce the same results under the same cost. So this will be cost-effective process. Means in which cost-effective, if we can calculate what is the cost-effectiveness of the drug of a delivery system, cost-effectiveness deals with all the therapy parameters. As such as in pharmacoeconomics field, we have discussed that what kind of costs are associated with any disease and application of a therapy. So a drug or a therapy that can produce effects in a minimum time and can make a person to be fruitful for the community in limited period of time will be of the best, will cause a best use of the therapy. So therapy will be cost effective only when the cost of therapy will be minimum, and every time the same at the same cost, same kind of therapy is achievable. So that type of therapy will be cost effective therapy. Just for example, we are in a process of mixing. If we are mixing two powders, one is a white powder and one is a red powder, both we should be sure that they should be of a same particle size. If they are of a same particle size, then we can mix them easily. Otherwise, different particle size will coagulate at different parts of the machine and they will not mix properly. For example, a white powder and red powder of the same particle size are to be mixed, then speed of mixing and the contact of the powder with the mixing agent is the primary feature which will be optimized. For example, if we are mixing it in a pestle and mortar, so the force of mixing or the rotation of the mixing is very low. And secondly, the pestle will shape the powder on the walls of the mortar. So the walls of the mortar will not allow it to turn back into the container. So it will take greater time, and if we optimize it based on the speed of the propeller, for example, if it is machine operated, the speed of propeller should be optimized at which it can transfer to the walls and walls can transfer it back to the homogenizer. So the speed at which the two-way movement, a movement towards the wall and from wall to the propeller is possible. At that speed, we can mix these powders within 5 to 10 minutes. And on the greater speed, from that speed, all the material can be tangentially falling on the wall of the mortar container or the container on the walls of the container. So mixing is not possible. And at lower speed, we will be confining it at the bottom, at the center, and it will be not be mixing on the side particles. So cost effectiveness will be if a process is optimized based on the speed of the mixing process will be optimized based on the speed of the revolution and the time of the mixing. So every time if the same time and same type of result is produced every time, then the process will be cost effective, as we will know how much time a mixing, a blending, a compressing, a granulation process, a quality control analysis, and its stability analysis will take, and at which time we can dispense that into a market, or we can send that into a market. Second goal is safe and effective medications. First of all, safety. Safety is the most important feature of any delivery system. Whenever we are administering a therapeutic substance, the therapeutic substance is known as a chemical entity. So a chemical substance is always toxic, and it shows dose-based toxicity. So dose-based toxicity should be reduced. So if we are giving a small dose, it will be eliminated from the body, as the elimination processes are first order, are depending on first order kinetics. Higher the dose of the drug in the biological system, higher will be its elimination rate. Lower the dose and lower will be its elimination rate. So for the safety purpose, we will have to adjust its doses. And secondly, we will have to remove its interaction and its incompatibilities. So incompatibilities can be of various types. For example, incompatibility can be a drug to drug. If two drugs are used, drug to drug interaction, or as we have blended it in a formulation with the excipient, so it could be drug to excipient interaction. So these could be incompatibilities with the body processes also. So if a medicine is free from any drug to drug and drug excipient interaction, then it will be called as safe drug. Any incompatibility can be a harmful or can produce a toxic responses when it is administered even as at a therapeutic doses. So it can cause maximum damages. So safety means a drug should be administered at a dose at which it is not toxic. And number two, in a delivery system, which is preventing its drug to drug interaction and preventing its drug to polymer interaction. Its chemistry should be known as polymeric chemistry is known. So we should choose only those polymeric substances for the delivery devices in which drug and polymer are compatible with each. So in that compatibility, it will be called as safe medicine. And if a drug is a therapeutic delivery system containing a therapeutic substance and excipient is considered safe, then it should be effective also. If state is achieved after administering a drug substance, then we can say that it is more efficacious results. Just for example, if a drug is administered at the therapeutic concentration into the biological system and its time of circulation in the body increases or it remains in the body more than greater time, then the need to administer a second dosage form, a second dose will be minimized or will be eradicated. So a drug will be more efficacious because it will remain at the therapeutic site for a longer period of time. So the efficacy demands that drug should be administered only and only at the dose at which it is at least above the minimum effective concentration. And secondly, the concentration is to be developed only at the therapeutic site. No need to develop a concentration in the whole body. So the drug's circulation time should be increased so that the drug should remain at the therapeutic site for longer period of time, and it should not go into any other part of the body so that it cannot produce side effects. So as the side effects will be reduced or drug delivery system will be called protected, so it will be more efficacious, or it will be known as safe and efficacious. And now proving my point with some examples why we need pharmaceutical technology, we will discuss in few examples. The first example are based on public crisis that appeared during the marketing of the drug products anciently in the past 15 to 20 years. First of all, chloramphenicol was actually introduced as an antibacterial in children in 1950s. That caused gray baby syndrome. Gray baby syndrome, in which a circulatory failure existing and the color of the body was appearing as gray or blackish. The blackish color appearing was indicative that circulation is not moving throughout the body, and that caused death. Syndrome actually caused respiration, myocardial respiration, and oxidative phosphorylation that caused the unexpected deaths in the children. So if we have developed an optimized chloramphenicol delivery system, it should not causing any kind of circulatory failure or circulatory collapse. So if at that time pharmaceutical technology will be present, this public crisis could be avoided. Similarly, in 2004, Merck introduced rofecoxib. Rofecoxib was introduced in the market by Merck in 2004, and it was a selective COX-2 inhibitor when used to mitigate the pain receptors. So when it was administered on longer term basis, it was seen that it is causing more decreased level of prostaglandin and thromboxane level in the heart. So that decreased level of prostaglandin and thromboxane level, PGI2, in the heart are causing the chances of heart attack. So heart attack chances were increased to double, which were using rofecoxib for longer period of time. So it was worldwide withdrawn in 2004 after 6 to 7 months of its post-marketing. Another example is Arthrotec. Arthrotec is containing diclofenac 50 milligram and misoprostol 200 microgram in a consultation. This is a combination of two drugs, diclofenac and misoprostol. Initially, only misoprostol was introduced into market as a PPI agent. So it could reverse the PPI response at 800 microgram doses. Means it is a synthetic prostaglandin that can cause PPI effect at 800 microgram doses. But as its half-life is very reduced, so we have to administer it repeatedly to achieve a dose of 800 microgram in 24 hours, depending upon its rate of elimination from the body. So when the dose of 200 microgram was taken after 3 to 4 hours for 4 times in a day, the dose reaches to 800 microgram, and that cause vaginal bleeding and termination of pregnancy in the pregnant women. So it was misused or abused to cause abortions. Similarly, a cough syrup which was introduced in China in 2005 or 6, they were using Chinese use traditional delivery of the system, and they are using some syrups to treat many ailments in the children and in the adults. So a cough syrup for children was actually for a cough syrup which was to treat cough symptoms in children, was prepared based on a solvent which is glycerine or glycerol solvent. Glycerol is a sweetener. Glycerol is secondly viscosity enhancer, and thirdly glycerol is antibacterial also. So glycerol was used as a solvent to prepare a syrup. In 2004 and 5, glycerine was not available, or a cheaper replacement of glycerine was introduced into market known as diethyl glycol. Similarly, propylene glycol. The first diethyl glycol was introduced, which was very of less cost than glycerine. So the company uses diethyl glycol instead of glycerol or glycerine. The diethyl glycerol is 100 times more potent than glycerine, and it will be 100 times more toxic than glycerine. Glycerols are actually compatible with body and not toxic, but when they move into tissues, they get solubilized and they can be metabolized within the tissues. So they can change the chemistry of tissues also, in which glycerine and propylene glycol are very much compatible with body, but diethyl glycol is not compatible. When diethyl glycol was added into the syrup, it caused nephrotoxicity and also multiple organ dysfunction in children. So multiple deaths of the children were reported. Now, secondly, in the second case examples, the first case example was based on safety of the drug substance. So the aim of the preparing pharmaceutical technology subject or devising pharmaceutical technology subject are safe and efficacious drugs. So along with safety, we should be very critical to efficacious system also. So we have developed some size-based delivery devices, or we have developed some mechanism-based delivery devices. Just for example, a disease which is known as cancer. So cancer is a disease which is only a defect of a tissue which was proliferating at a normal rate, and now it has been converted to an uncontrolled proliferation rate. The same process is happening all over the body. All body cells are actually replicating, and our stem cells or bone marrow cells are replicating at a higher rates. So if any of the cell is replicating uncontrolled, so that will cause cancer. And if we are administering a drug for that cancer therapy, it will pass on to all the body cells which are actually replicating. It will specifically move into the bone marrow cells, which is producing red blood cells, which is producing factors, which are producing all the defensive mechanism, lymphocytes, every kind of the products which is produced by bone marrow cells. The drug will be moved into the bone marrow cells, and bone marrow cells, the production of the cells will be inhibited or will stop, and on inhibition of the bone marrow cells, new cells will not be prepared. And if red blood cells are not prepared, white blood cells are not prepared, red blood cell cannot transfer oxygen and nutrition, and white blood cell cannot produce immunity to the foreign particles. So administering an anti-cancer drug to a patient with a cancer, we can define the death period of the patient also, because it will cause cessation of the normal proliferating processes, and that cessation will cause multiple type of diseases, multiple type of infections, and gray baby, as there was a gray baby syndrome. So deficiency of movement of oxygen and iron into the body parts. So for that, we have a study that whenever a cell has an uncontrolled proliferation rate, it causes more pores on the cells, so that more liquid can be absorbed and more wastes can be eliminated from the cells, so that a faster rate of proliferation can exist. In this way, the cancerous cell will develop new type of a vasculature, and that vasculature have a more pore size than the normal size of the body, even the more pore size than the eliminating organ. And if we prepare a size-based delivery system that have a size greater than the normal proliferating cell pore size and greater than the our eliminating organ pore size, for example, glomerular filtration, then it will not be eliminated from the body. It will remain within the body for the longer period of time. So we have developed some size-based delivery and some vesicular system that can hold a drug. Liposomes, niosomes can hold a drug within the compartment, and they can be coated with the inert polymeric substance, for example, polyethylene glycol. And those liposomes will be known as pegylated liposomes. And that pegylated liposomes have a greater size, and they can only move in a cancerous cell with having a greater pore size, and they cannot go in any parts of the body. It is very difficult to devise or to determine the pore size of the cancerous cell, but it is easy to determine that how a cancerous cell is taking some nutrients and how it is causing circulation to dry towards itself. And if we can find the process in which the medication, in which the nutrition and oxygen is moving or circulation is moving to the cancerous cell, we can deliver according to that circulation our delivery system into that cancerous cell. So for that, as an example, if the cancer is of the lower sides, for example, ovarian cancer or prostate cancer, we have found that in ovarian cancer and prostate cancer, there are multiple type of folate receptors which appear on the ovarian surface and also prostate surface. The overgrowth of receptors on the ovarian cells and on the prostate cell will allow us that we can cause a ligand of drug with the or delivery system with folic acid. So folic acid will be moved only to the folate receptors, and the maximum folate receptors are present on the ovarian surface or cancerous side. So the delivery system will move throughout the body and will be attracted towards the folate receptors. And when it will be attached to folate receptor, folic acid will be removed from the delivery system, and delivery system will be taken up inside the cancerous cell. And the anti-cancer drug will cause action in the cancerous cell only. So in this base, we can achieve targeting. Similarly, we have found that there are many diseases which cause pH, which cause specific pH of that area at which disease is present. If we are administering a drug substance with a coating of the cationic or anionic polymer, which are of the category which are polysaccharides, generally polysaccharides. So polysaccharides are not metabolized within the systemic circulation. They will move only and only to the side because it is a pH-based pH-sensitive polymer. So when they will reach at the specific pH, at that pH the polymer will dissolve, and the drug will be delivered at the site at which the polymer is dissolved. So two types of the drugs. One the drug which shows selective absorption at a specific pH, having a isoelectric point at the pH, or secondly, a disease site having a specific pH. They can be co-administered by the technique of site-based delivery using pH-sensitive polymers. Now, for example, insulin, which have a dielectric point of 5.4 to 5.8, that can be administered orally instead of subcutaneous injection if we coat it with a polymer that can deliver it to the large intestine in the ascending colon. The drug will only be released or insulin will be secreted only when the delivery system will reach at a pH 6 or pH 5.8, that is at the ascending colon. And when it will be released at the ascending colon, as dielectric point of the insulin is 5.4, the pH of the ascending colon at the center point almost is 5.4 to 5.6. So at that, it will be maximally absorbed to the systemic circulation and in the same quantity as we are subcutaneously administer some units of the insulin. Similarly, you can orally administer these insulin by using pH sensitive polymers. Next, the last one is reducing side effects. How we can reduce side effects? Actually, side effects we are saying are due to the higher doses, because we have to maintain a therapeutic level for longer period of a time. For that, we actually administer repeatedly a therapeutic substance repeatedly. So on repeated administration, the drug can make a therapeutic response, but it will cause side effects also. Just for example, doxorubicin, when it is given at higher doses, it causes severe side effects of cardiac side effects. So doxorubicin requires that this cardiac effect should be minimized. This can be minimized by encapsulating doxorubicin in our delivery system that can cause permeability and retention. It can cause permeability of the drug within the therapeutic area or diseased area, and it will retain the drug for longer period of time also. So it will be known as EPR effect for passive administration of any drug substance. So drug will be accumulated within the diseased site only, and it will not cause cardiac effects because it cannot move to the cardiac cells when we have only enhanced its permeability to the cancerous site, and we have enhanced its retention only in the cancerous site. So it will cause reduced side effects. Similarly, amphotericin B is an antifungal drug and can be used to treat serious and life-threatening fungal infections. We require higher doses of amphotericin B to cause fungal inhibition. So higher infusion rate is required. When we increase the rate of the infusion amphotericin B, it causes cardiac arrhythmias, which is also a life-threatening process. So to treat a life-threatening disease, we are actually producing a side effect, which is also a life threat. So amphotericin B's use should be minimized. For this, if we are using some delivery devices which are vesicles, for example, liposomes or niosomes, if we administer it into liposome or niosome, we can increase its rate of injection, infusion. It will not be moved to heart because it will move into heart, but the liposome will protect it, protect amphotericin B to be released within the heart. It will only allow its release when it will move into the fungal surface due to the liposomal interaction with the fungal surface. On interactional basis, it will release amphotericin B. So the cardiac arrhythmias can be reduced. Jazakallah wa ahsanal jazaa.